Hello students, welcome back to my biology online classes. In my previous lectures, I had discussed with you about cell surface receptors and its types and examples. That class I have discussed about three major types of cell surface receptors. The major family of cell surface receptor is Gpcr. Full form of Gpcr is G protein coupled receptor, which is our point of interest today. This Gpcr and G proteins, they are largest family of cell surface receptor. G protein coupled receptor. G protein coupled receptor. Now let's concentrate what is this G protein. The full form of G protein, which one is very much important abbreviation. The full form of G protein is guanine nucleotide binding protein. Guanine nucleotide binding. Now let's concentrate how Gpcr acts. Gpcr transmit signals via intracellular target. All of we know that the cell surface receptor, in case of cell surface receptor is present outside the cell, and this type of receptor cannot, this type of ligand, they are hydrophilic in nature and normally they cannot pass the PLP layer of plasma membrane, and in this case they are carry inside the cell with the help of this cell surface receptor after binding. So that Gpcr transmits signals via their intracellular target, means G proteins, their target, what is their target? Target to reach inside the cell intracellular. That their target is situated or located inside the cell. So G protein transmit their signal via intracellular targets. And this G protein, this is present in all eukaryotes. G protein, it's a very common type of receptor present in all eukaryotes. Note this point very carefully. Gpcr is present in all eukaryote cell, and Gpcrs are receptor. Take some example. Who are receptors of Gpcrs? The receptors of Gpcrs are neurotransmitters, neuropeptide and peptide hormones. Neurotransmitter, neuropeptides, peptide hormones. These three are main receptors for Gpcr: neurotransmitters, neuropeptides and peptide hormones. And Gpcr family includes a large number of receptors that are responsible for light, smell, taste. Now let's concentrate about structure of Gpcr, another very important feature. Gpcr consists of a single polypeptide. Gpcr consists of a single polypeptide of multipurpose nature, and they cross lipid bilayer 7 times. That's why it's called serpentine receptor. Note this point very carefully. Gpcr cross lipid bilayer means protein, lipid, protein, that lipid bilayer of plasma membrane. Gpcr cross that lipid bilayer of plasma membrane 7 times. That's why this receptor is called serpentine receptor. An example of Gpcr response to chemical signal is beta adrenergic. And this beta adrenergic receptor binds to beta adrenaline hormone and we are very familiar about this adrenaline hormone. Adrenaline, not beta adrenaline, adrenaline or epinephrine hormone. Normally, GPCRs are linked to trimeric G protein. GPCRs are linked to trimeric G protein. GPCRs are linked to trimeric G protein. G proteins are GTPase. This one is very much important conception. GTPase switch protein. All of we familiar about ATP. ATP, GTP, CTP, TTP all are comprises about 3 inorganic phosphate. Okay, 3 inorganic phosphate. That's why this one is called triphosphate. 3 inorganic phosphate in that terminal end. See, this ATP, from this ATP, one phosphate is released and energy is released, and this energy is utilized when necessary. Okay, and this triphosphate now change into ADP. ADP plus PI and release of energy. In case of ATP, we know. So same things occurs in case of GTP also. When needed, the one terminal phosphate is released, energy is also released, and this triphosphate GTP, when it is GTP, the triphosphate is released and GTP becomes to GDP. And in case of G protein, when it's bind to GTP, they are active, and when it's bound to GDP, normally it remain bound with GDP. Normally not all time activity is needed, all of we know. So normally they are bound with GDP, which is an inactive form, and when needed, when signal is coming, ligand is coming, and its work mode is on, that means it will go to bind with GTP. Got it? GTP is switch protein, and there are 2 classes: trimeric G protein and monomeric G protein. And G protein turned on when bound to GTP, and G protein turned off when bound to GDP. G protein turned on when bound to GTP, and G protein turned off when bound to GDP. Normally, in the absence of a signal, G protein is bound to GDP. Signal is coming from outside. Signals activate the release of GDP. And what is the scenario of normal cell? Normally in our inside our cell, there are lots of GTPs are present inside our cell, and when signal is coming, what happens? The subsequent bind to GTP over GDP. Got it? One GDP is already bind with in its inactive state. GDP is already bind with this G protein. Normally, when signal is coming inside our cell, there are lots and lots of GDPs are present inside our cell. Okay, and when signal coming, what occurs? The subsequent release of GTP over GDP is favored by the high concentration of GTP present inside our cell. The intrinsic GTPase activity of this GTP hydrolyzing protein, of this GTP binding protein, hydrolyze the bound GTP into GDP plus PI. Okay, I have already shown here GDP plus PI. PI means inorganic form, thus converting that active form into inactive form again. Now let's concentrate about structure of G protein PLP layer. this structure should be drawn here. I am writing here for time saving as straight line, and this is figure number A. This is protein, lipid, protein of plasma membrane. This is plasma membrane. This is extra cellular space. This is cytosol. Let's draw G protein. I have already discussed with you that it is going to cross plasma membrane 7 times. The serpentine nature, 7 times it will going to cross lipid bilayer 7 times. This will be a 3D diagram. This one is Gpcr. And as this is protein in nature, except one amino end, NH2, and carboxyl end. Amino end is present at the cellular space, and this carboxyl end is present inside the cytosol. Here, this is Gpcr, and now I am going to draw G protein, which is also attached to this membrane. Alpha, beta, and gamma. GDP is bound normally. It's inactive form. GDP is bound here. Inactive G protein. So let's concentrate about structure of G protein. G protein has 3 subunits. G protein has 3 subunits: alpha, beta, and gamma subunits. Okay, these alpha and gamma anchored to membrane lipids covalently. Note this very carefully. Alpha and gamma attached to membrane lipids covalently. Anchored to membrane lipids covalent. And this beta subunit, it always bind with gamma subunit, and this will go to form heterodimeric structure. Heterodimeric structure. This alpha subunit, this one is GTPase switch protein. So main function of G protein performed by alpha subunit, as it is GTPase switch protein. This helps to alternate with active GTP form and inactive GDP form. When bound with GDP, it remains inactive. When bound with GTP, it becomes active. Binding of a trimeric G protein plus activated receptor. What happens? Dissociation of GDP, then binding GTP and G alpha, and then dissociation of G alpha GTP from G beta gamma. This is the scenario of G protein step by step. Now let's draw here all these steps. Now let's draw here. I am not going to draw in detail. This will be 3 steps diagram, A, B, C. Please draw like this in all these 3 cases. Now let's step B. This is plasma membrane, PM, and here Gpcr. Let's draw a rough diagram. Okay. Here NH2 end, and from here COOH end. Now ligand is coming. Normally this structure, now ligand is coming from outside. Ligand is coming and ligand is bound to this side. Okay, this one is G protein coupled receptor. ligand, now ligand get binding, and here it will be activated. It will be activated. Okay. Ligand bind here from extracellular space, and here its activation is started. And here G protein is present, alpha, beta, gamma, and with GDP inactive form. Now what happens? Here GTP to GDP. This one is very much important, note it. For accumulating space here, I am going to erase this. Here step number 3. Also PM, and here okay, ligand is here, ligand is here, ligand binds here. This part is activated. Okay, alpha, beta, gamma. Here GTP is bind now, GTP, okay, and it being activated. Here this one is now activated, alpha, activated alpha subunit, and here activated beta gamma complex. This figure is very much important. This 3-step figure should be drawn when questions about Gpcr is coming. This picture is very much important. This figure is very much important along with its description. But without this pointing and without this diagram, this answer is not properly right and not properly being described. So be careful about this figure. G alpha GTP and G beta gamma this one can specifically interact with effector protein, effector proteins. Okay, this leads to intrinsic GTPase activity of G alpha. This inactivates, all is set, all is done, activity is over. Then came to that inactive condition. This inactivates G alpha GTP by catalyzing GTP hydrolysis. Then what happens? G alpha GDP then dissociates from its effector and reassociate with G beta gamma. G alpha and GTP then bind to effector protein. Next, intrinsic GTPase activity of G alpha. Then the next step, activation is done, all set. Okay, then inactivates G alpha GTP by catalyzing GTP hydrolysis, and after GTP hydrolysis what will be found? GDP will be found, and G alpha GTP then dissociate from its effector. It's no more bind with its effector, all its function is done. Then reassociation is with G beta gamma. Okay, this is all about how G protein functions.